So our next section of this unit is about genetics. And in 7th grade, we had a unit on genetics. We talked about some sort of basic inheritance and Punnett squares, so that should be familiar to you. So in this year, we'll go into some more detail about some more advanced genetics, a little bit with multiple alleles and codominance, and talk a little bit more about the environmental effects on gene expression, some things like that. But just to sort of review, genetics is the study of how traits, how characteristics are inherited from parents to offspring. And this happens through the passing on of DNA in the form of chromosomes. Gregor Mendel's often named as sort of the father of genetics. And we talked about him a little bit back in 7th grade. Mendel was a monk, an Austrian monk, religious person. He was also a gardener, hobbyist gardener. And he was growing pea plants. He would crossbreed them together. Pea plants, you can easily pollinate one to the other with a little paintbrush. And so you take pollen from one pea plant, transfer it to the stigma of another pea plant, grow the fruits, grow the seeds, and see what the offspring were going to be like. And he did this thousands and thousands of times. Mendel kept very careful notes. He had large amounts of data. And as he was doing this research and recording this data, he started to see patterns emerge. Patterns in how traits were inherited, and which traits were displayed in the offspring and which traits were not. So he came up with some laws that we're going to talk about today. He came up with some ratios when certain parents produce offspring that he continually observed. His research was kind of almost forgotten for a while. He was doing his work in the middle of the 1800s. This is before anyone knew anything about DNA, okay, or genes. He didn't know the word gene. He didn't use the word gene or allele. He called them factors. But his work was sort of rediscovered later on in the 20th century. Thomas Morgan was an American geneticist who sort of repopularized some of Mendel's work and confirmed that what Mendel observed in these pea plants also applied to animals. Morgan's main organism of research was the fruit fly. In fact, fruit flies are still used today in genetics research. If you take intro biology in college or a genetics course, you may work with fruit flies. Fruit flies reproduce really quickly, so they can go through generations, and you can observe how traits are inherited. They have several, their chromosomes and the genes present are well known, so they're still commonly used. And then a major advancement was the discovery of the structure of the DNA molecule, how it's actually composed. Its three-dimensional structure was found by Watson and Crick with help from Rosalind Franklin. Watson and Crick received the Nobel Prize for putting together this model which showed that a DNA molecule is a double helix with nitrogenous bases connecting in the middle, and then the sides are composed of sugars and phosphates. And since then, we have learned quite a bit about DNA to the point where today we can edit DNA in organisms. And we'll talk more about that towards the end of the section. So we do need to review DNA a bit. So we talked extensively about it in biochemistry. We talked about DNA, we talked about RNA, we talked about protein synthesis. So just to sort of refresh our memory about that, we know that the DNA molecule, it's a nucleic acid, one class of organic molecules. And DNA is composed of two strands. And the sides of, when they're bonded together, the sides of the ladder, if you want to call it that, are phosphates and sugars bound together. And then the rungs of the DNA ladder are made of the bases. DNA has 4 different possible bases: adenine, thymine, cytosine, and guanine. And we abbreviate them A, T, C, and G. And we know that they are complementary to each other, that bases match up in a predictable way. Adenine and thymine are complementary. Guanine and cytosine are complementary. The 3-dimensional structure is this spiral staircase, this double helix. And DNA can replicate itself because as the DNA molecule unzips, new bases, new nucleotides will fill in on each side. And because they're complementary to each other, the end result are 2 identical strands of DNA. So as we just talked about meiosis and we earlier talked about mitosis, the first step in interphase is the chromosomes need to make copies of themselves, the DNA copies. And it does that through this process of DNA replication. We take one DNA molecule and we end up with 2 identical molecules after replication. The way that DNA actually influences traits is because the DNA molecule, the order of the bases, the sequence of A's, T's, G's, and C's, is a recipe or the instructions about how to build a single what? Not a cell. One gene is the recipe to build one protein. And so genes control the synthesis of proteins. And this slide sort of refreshes how that happens. So remember, DNA is in the nucleus. It's stuck there. It can't leave the nucleus. It has in the sequence of bases the instructions about how to build a protein. So when that protein needs to be synthesized, the DNA in the nucleus partially unwinds, and a copy of that recipe is made in messenger RNA, another type of nucleic acid. Messenger RNA is similar to DNA, but it's only one strand, and it doesn't have T in it. It has U instead, uracil. So that messenger RNA gene, it is able to leave the nucleus. So after the recipe is copied to messenger RNA, it leaves the nucleus and goes out to the site where the protein is going to be made. What is that site that makes proteins? Ribosomes. They're made of RNA as well. So the recipe is brought out to the ribosome. Every 3 bases on the messenger RNA is a code for 1 amino acid. How many possible amino acids are there? Remember that? No? Less than 32. What? Less than 22, but you're getting close. There are 20 amino acids, which are put together to form all of the different proteins that we need. And so the messenger RNA bases tells which amino acids to put together. Every 3 messenger RNA bases is a codon for 1 amino acid. Transfer RNA molecules bring in the proper amino acids. They start to get linked together until the protein is made. Remember, a protein will contain thousands often of amino acids linked together. Once they have been linked together properly, it's released. It takes on its specific shape, and it can go and do whatever function it needs to do. So that's sort of summary of protein synthesis. All the details are back in biochemistry. We need to look that up, but the two steps are called transcription and translation. So transcription is here when the RNA is made, the messenger RNA from the DNA. And then the second step, translation, is taking that recipe and actually putting the amino acids together to form the protein. All right, so let's talk about Mendel a little more. Mendel was studying pea plants. Pea plants were sort of an excellent organism for him to do this research because they have very clear traits. You look at a pea plant, the seeds are either smooth or they're wrinkly. They're yellow or they're green. The pods are yellow or green. The flowers are white or purple. So they have very clear, very distinct traits that come in just two versions. And so when Mendel was doing his research, he was studying these plants and taking very careful notes about, okay, if I take plants that are pure purple flowers and I breed them together with pure white flowers, here's what I get. You know, what percent of the seeds that were growing showed purple or white flowers. So he studied and kept careful notes of all of these characteristics. He came up with some rules, some laws, we call them. And so the first is the law of dominance. And the law of dominance says that some alleles, some versions of the gene are dominant, while others might be recessive. And so if an organism has one of the dominant alleles, that's what's going to show through. The dominant allele. Typically, when we're completing Punnett squares, we use a capital letter to illustrate dominance. So if we're writing an allele with a capital letter, typically that means it's the dominant version. And we use a lowercase letter for the recessive version. And so one of the things that Mendel saw was that when he had pure tall plants, pure tall pea plants, and he bred them with pure short pea plants, and he looked at the offspring that were produced, they weren't a mix of traits, they weren't medium height. He found that they were all tall. And that's because for height of pea plants, tallness is the dominant allele. So if we do a Punnett square showing the tall parent, being crossed with a short parent, we fill in the boxes to see what the offspring would be like. And what do we see in this case when you fill it in?
Dominant gene, recessive.
Yeah, all of the offspring are the same. They all inherit a dominant allele from one parent and a recessive allele from the other parent. That's the only possibility for this trait. So let's just review the terms we use. So when we talk about how the genes show through, what they actually do in the organism, we're talking about the phenotype, the physical traits in this example. It's the result of what the genes actually do. When we talk about the genotype, we're talking about the alleles present in this organism. When we have two of the same allele, we call it homozygous. When we have two different alleles, we call it heterozygous. So these are a homozygous dominant parent crossed with a homozygous recessive parent, and they form 100% of the offspring being heterozygous. Mendel called the original purely bred generation the parent generation, the P1 generation. The resulting offspring he called the F1 generation. Okay, so these are the F1 offspring. Now, if we take two of these F1 offspring and breed them together, we get the F2 offspring. So what do we get in this box? Give me the technical terms for this. Grady, what do we get here? Homozygous dominant individual. What would phenotype be, Grady? Tall or short? Tall. All right, what would the genotype of this offspring be? Somebody else. Carter?
Heterozygous.
Heterozygous. What would its phenotype be, Carter?
Tall.
Tall. How about down here? Genotype, Bella? Heterozygous, phenotype? Tall. And in our last box here, Zach?
Homozygous, recessive.
Yep. And that would be a short plant. And so Mendel saw this repeated with flower color and wrinkled or smooth peas and green or yellow peas. He saw that whenever he bred together two heterozygous individuals, he ended up with this ratio of 3:1. 3 of the organism would show the dominant phenotype and 1 would show the recessive phenotype. And that led him to start to think about why that would be the case. And so we have a couple other laws that Mendel talked about. One is the law of segregation and recombination. So let me go back for a second here. So when we have this parent, when it produces gametes, what happens to these two? So this big T allele is on one homologous chromosome. This little T allele is on another homologous chromosome. And what happens in meiosis to homologous chromosomes? Well, they could cross over, and then eventually they get separated from each other. And the gametes contain one from each homologous pair. So when this offspring is producing this parent is producing gametes, some of the gametes will get this homologous chromosome with the big T allele. Others will get the little t. Half will get the big T, half will get the little t. Same thing when we have this offspring produced. And so that is the law of segregation and recombination. That when the gametes are being formed in meiosis, the homologous chromosomes get split up. And each homologous chromosome is carrying one of the alleles. That's what's being shown here. One parent has big T, little t. When it produces gametes, some of the gametes get a big T, some get a little t. The alleles have been segregated. When do they recombine? When do these gametes recombine? What do we call it? Is that?
Fertilization.
Yeah, when fertilization happens. Two of these gametes are going to come together in fertilization, and then we'll have two alleles. Which gametes meet in fertilization determine which genotype we end up with at the end. The law of independent assortment says that if the alleles are on different chromosomes, they will be inherited separately. So in this example here, let's look at this. So if we have brown eye hair color and eye color. Okay, we have brown eyes, blue eyes, we have black hair, red hair. So if the chromosomes line up one way, okay, you end up with certain combination of those characteristics. But if they line up the opposite way, the gametes get different combinations of those characteristics because each of those alleles is on a separate chromosome. What do you think it would mean if two alleles were on the same chromosome? What if flower color and height of the plant were both on one chromosome? What do you think that would mean about those two traits? Come on. That they're linked and they would be inherited together. That if you got purple flowers, you would have a tall pea plant because they're on the same chromosome. Do you know what could switch that around? How could they be separated from that chromosome? Yeah, crossing over could unlink them. One thing that Mendel had to do was have a way of figuring out. So if purple is the dominant allele for flower color, if you have a pea plant with purple flowers, well, you can't be sure of its genotype because there's two possibilities. It could be homozygous dominant. That would result in a purple flower. Or it could be heterozygous. That would also result in a purple flower. So just looking at the flower, you couldn't be sure of the genotype. But you could figure it out by completing what's called a test cross. You have an organism with the dominant phenotype. If you cross it with a pure recessive, a homozygous recessive plant, you could look at the offspring and figure out what the parent was. Because if you take the purple parent and cross it with this homozygous recessive flower, if you get some offspring that are white, what does that mean about its genotype? Was it this or this? If the offspring turn out to be, or some of them are white. So which one? If the genotype is this, will you ever end up with white offspring? No. So if you do this test cross, you cross this dominant phenotype with a homozygous recessive plant. If some of them turn out to have the recessive phenotype, then you can be sure that the parent was heterozygous. If you do this and none of them come out to be white, then you could be relatively confident that it's homozygous dominant. Now you can do Punnett squares for multiple traits at one time. We call this a dihybrid cross in this case, because we're looking at two different characteristics. We're looking at whether the seeds are round or wrinkled. You don't have this slide. And whether or not they are yellow or green. The procedure is basically the same. It's just a little more complicated. You have a 4x4 Punnett square rather than a 2x2 Punnett square. What you do is you write all the possible combinations of gametes from each parent on the top. So for example, we have a parent that's big R, big Y, little y. The possible gametes you can have, you can get a big R and a big Y. You can get a big R and a little y from this parent. Or you can get little r, big Y, little r, little y. So there's 4 possible gametes. Same is true here. Fill in our Punnett square the same way, but we have 16 squares and we can look at the results to figure out the likelihood of getting any combination of those traits. Now, Mendel's traits that he was looking at were pretty simple. They were either dominant or recessive. They were all inherited independently. But there are more complexities than what Mendel observed in his pea plants. And intermediate inheritance includes some of these complexities. In intermediate inheritance, the heterozygous individuals are different than the homozygous individuals in some way. As we talked about, in the purple white flowers, the heterozygous plants look the same as the homozygous dominant plants. But in intermediate inheritance, that's not true. One example of this is incomplete dominance. In incomplete dominance, heterozygous individuals sort of have a blend of traits between the dominant and recessive. So one example are these snapdragon flowers. So the snapdragon flowers, if you have a pure red flower that is cross-pollinated with a pure white flower, what you end up with are actually an in-between color, a pink-colored flower. The heterozygous flowers are halfway between the other two alleles. And so often if we're showing incomplete dominance, we use capital letters, but we use different letters. For example, the snapdragons that are pure red are big R, pure white are big W, and the pink ones are RW. These Andalusian chickens have a similar type of inheritance. If you have a black chicken and a white chicken and you breed them together, you actually get this grayish colored chicken. They have incomplete dominance. Another version of this is codominance. This is when both alleles are expressed at the same time. For example, in these roan cattle, okay, if you cross a red roan cattle, a pure red, and a pure white roan cattle, what you end up with is actually a spotted cattle that has some parts which show the red phenotype and some parts which show the white phenotype. Okay, again, we can combine those letters to show that. Another type of inheritance is multiple allele inheritance. So everything we've talked about so far has been like two alleles, purple or white flowers, tall or short pea plants, wrinkled or smooth seeds. But there are many traits which are more complex than that. They're actually determined by more than two alleles. Human blood type is an excellent example of this. There are actually 3 alleles for human blood type. There's the A allele, the B allele, and the O allele. And so any individual gets 2 out of those 3 possible alleles. If a person gets the 2 A alleles, they have type A blood. Pretty simple. If they get an A allele and the O allele, they still have type A because I is recessive. The type O allele is recessive. Same thing with type B. They get 2 B alleles, they get type B blood. If they get a B and an O, they're still type B blood. If they get an A and a B, that's what gives type AB blood because both of these alleles, they're codominant, so they're both expressed. The only way a person ends up with type O blood is if they get 2 of the O alleles. And so we have 3 separate alleles which determine human blood type. We can do a Punnett square showing this. If we have a person that has type AB blood crossed with a person with type O blood, we can fill in the Punnett square in the same way. We see that 50% of their offspring have type A blood and 50% would have type B blood. This can get much more complicated than that even. Rather than having just 3 alleles, okay, we could have 4 different alleles like we do in these rabbits. So these rabbits have all of these different alleles. Some are dominant over others, some are not, and they can inherit them in all of these various combinations. And that leads to these rabbits having all of this large variety of fur color, from albino to dark gray to light gray and so forth. So that's controlled by 4 different alleles that dictate fur color. All right, any questions up to that point? We're going to stop there for today in our notes.